Thank you everyone for joining us today. Dr. Collins is going to go ahead and cue up his slides. Let me welcome you to the second of the NIH OITE Summer Lecture Series. It has been a really special summer, having so many interns working creatively using virtual platforms and resources, yet connecting and making their presence felt on campus. I know we're joined today by students across the United States doing internships at other institutions, and we welcome you as well. I hope that you and your families and loved ones are well. We are certainly turning many corners in our sense of COVID safety, but of course, I appreciate that many of you have family and friends across the globe, and there are still many, many struggles ahead. So we wish you all the very best for all of them. It is really an honor for me to turn the Zoom floor or Zoom podium over to Dr. Francis Collins, who really needs no introduction. And in fact, he'll talk about himself and his journey and his career in his talk, so I won't spend time on that. But I do want to say one thing by way of introduction. Dr. Collins, as you can imagine, has been unbelievably busy before the pandemic, but especially in the last 14 or 15 months. Yet every time we ask him to do something with trainees, he says yes. I know, because I work with people in his lab, that he has been available for them even through all of these hectic times. He is really a mentor and champion for training. So I know that you all join me in welcoming him. Dr. Collins is going to give a short talk and then take questions. You can put them in the Q&A. Please tell us where you're from, what education level you're in at the start of the question. People will be able to then upvote questions that they'd like to see answered, and then I will do my best to pick some questions from a variety of different areas. So thank you, Dr. Collins, for joining us.
Oh, thank you, Sharon, and thanks for all you do to lead our Office of Intramural Training and Education, which now seems to be extending itself well outside of the NIH to lots of other places. And I'm delighted that all of you have joined up for this conversation. Well, sorry, I'm a couple minutes late. I was just in an intense meeting with the FDA talking about a gene therapy protocol for a disease my lab has worked on now for 20 years, the rarest form of premature aging called progeria, where we have reached the point where we think we are about ready to try a clinical trial of in vivo gene editing to fix that single nucleotide that causes this disease in children with the illness. And it's pretty amazing to be able to say that. So kind of all a buzz from that experience. And that is indicative of the kinds of science that now have become possible in ways that I don't think I would have imagined would happen this soon. And so all of you who are working in various areas of biomedical research, you have arrived at a really good time for the potential here of science, even though it may feel like you arrived at a really bad time because of COVID-19, which has been such a disruption for all of us in the course of the last year and a half. So I thought it would be useful before we get to the Q&A for me to give you a little bit of an autobiography and maybe some comments, a little philosophy about the kinds of things you might think about as you're approaching your own decisions about how you want to travel an exciting path in medical research. You may agree or disagree with my recommendations, but hey, Sharon thought that would be useful as well. So I'm assuming you can see my slide, and hopefully it is filling the appropriate space. And so if we go on to the next one. Okay, I'm starting a ways back here. Yes, that would be me in the left at about age 8, on the right at about age 16, trying to look like somebody that people would want to listen to. Yeah, I grew up on a farm in the Shenandoah Valley. Very much a wonderful experience as a kid, although I don't know if I totally appreciated it. Of being in an environment with a lot of music and theater. My parents were theater people. Although we lived on a farm, we weren't very successful at that part. And I was homeschooled till the 6th grade by very talented parents who felt that they could do a better job than the local schools. And with me, they really, I think, achieved that, in that they taught me to love learning new things. And I hope that's something you all can resonate with. I got into public schools eventually and fell in love with chemistry in a 10th grade class and figured, well, that must be what my calling is. I'm going to be a chemist. And so I went off to University of Virginia as an undergraduate, and I majored in chemistry, and I ignored biology because it seemed messy and focused my efforts on chemistry and physics. And having enjoyed that for 4 years at UVA, I figured, well, okay, I better go get a little more training. So off I went to get a PhD in physical chemistry at Yale. That was an experience also that was challenging and interesting. And my project was really about quantum mechanics. And a lot of it was done with pencil and paper, trying to solve second order differential equations. Yeah, that's me trying to look as if I knew what I was doing. And you can see the title of my PhD thesis there, which was very much a theoretical project. I didn't touch any instruments except for computers. And they were pretty rudimentary at the time. But along the way, I kind of realized maybe I've narrowed my horizons a little bit here by being so focused on the really basic things in physics and chemistry. You'll notice the title of my PhD thesis got no further than protons and hydrogen gas. Probably there's some other molecules that are pretty interesting. And then I discovered this one. This was because there was another grad student I was friends with who was studying DNA in that same department of chemistry at Yale. And the more he told me about what was happening, recombinant DNA was being invented. Things were happening here that could be profound in terms of our understanding of life and how it works. And I began to realize, hey, life has principles too. This could be really an interesting area of science, and maybe that's what I want to do. Without having any better notion than that, I figured, well, okay, time to change my trajectory. And this was pretty wrenching because, already well into a graduate degree, I was already married, I had a small child. I decided it's time for a change. And as long as I'm going to change into life science, I want to keep all my options open in case this is something that could even be applied to human beings in a medical sense. So off I went to medical school at the University of North Carolina. I was a little worried about whether that was going to work out, but it worked out almost immediately. That I realized this is science, but it's about the human body. It has human applications. And wow, it's about DNA too. DNA is in there. And I was pretty certain I wanted to try to bring genetics and medicine together. And in many ways, that's kind of what I've been trying to do ever since. After training as an internist, after medical school and residency, I realized I needed to know the science of this better. And what do you know? I went back to New Haven, this time as a fellow, studying molecular biology and learning the clinical experiences in medical genetics that allowed me to have that as my subspecialty. And then finally, after all of this, you might wonder, does this guy really want a job or does he just want to keep training? I ended up as a faculty member at the University of Michigan, an assistant professor, doing what you do, taking care of patients, teaching medical students, and running a research lab. And my research interests were to try to take the tools, limited though they were at that time, of genetics and come up with answers to diseases that we really didn't understand at all. We knew they were inherited, but we didn't know what gene was involved. And that was thousands of diseases, and particularly some of the more common ones. And so I became a gene hunter, basically trying to take what we could do in terms of traveling around the genome with very clunky tools. There was no genome project. There was no internet, really. It was really hard going, and try to see, could you find for certain disorders where you knew it might be as subtle as one letter misspelled out of 3 billion, could you actually locate it? The picture you see on the right was my justification for why this was hard after we'd been spending years trying to track down the cause of cystic fibrosis, and people wanted to know, well, do you have it yet? This is me holding up a needle, sitting in a Michigan haystack saying it's really hard because that's the scale that we're struggling with here. But ultimately, thanks to a lot of really hardworking folks in my lab and a collaboration with a wonderful investigator in Toronto, we were able to identify the gene for cystic fibrosis in 1989. This was years of work, Something that today, probably any one of you, given some DNA samples from families, a thermal cycler, and access to the internet, you could probably do this in a week. For us, it took about 6 or 7 years, and probably at least 100 scientists along the way working, many of which ultimately went on to do other things because this was so frustrating. So we knew that that was not going to be the solution in the long term, which, by the way, was one of the motivations for the Human Genome Project. Along the way, I had the chance to work on other disorders that cause human illness, Huntington's disease. That's Nancy Wexler, colleague that I worked with along with other researchers and ultimately found the gene for Huntington's back in 1993. Neurofibromatosis, a relatively common, dominantly inherited condition, which affects kids and adults, which we also were able to track down. And most recently, my lab's been working on this rarest form of premature aging that I just mentioned, having had this meeting with FDA a few minutes ago. But then what happened? Well, what happened was, here I was at Michigan being quite happy, but the Human Genome Project got started. And I believed strongly that that was going to be an important way to make this kind of gene discovery happen much more efficiently for the thousands of diseases that were still waiting for that kind of advance. But I thought I could manage my part of the Genome Project from Ann Arbor. And so was delighted when none other than Jim Watson, here on the right, stepped in to be the first leader of the Human Genome Project and he was hired to do so. But then something happened in 1993. On the left, you see Dr. Bernadine Healy, the director of the NIH at that point. She had the job then that I have now. And let's just say Dr. Healy and Dr. Watson did not get along. And Dr. Watson was not known for being diplomatic. In fact, quite the opposite. And after a while, it became untenable. And she basically said, okay, you're out of here. And then she had to find somebody else to come and lead this fledgling effort. The Genome Project was just barely getting off the ground. Most people in the scientific community weren't convinced it was a good idea. But she calls me up and says, I want you to consider coming to NIH, becoming a federal employee, and leading this uncertain project that's kind of sprawling all over and doesn't have technology invented yet that's going to make it possible to succeed. Are you interested? And I listened and I said, boy, that's a great honor because this is going to be a really remarkable project, but I don't think so. A few more months go by. She reached me out to me again. And I realized that over those months, I had really been struggling about how I could turn down something that might be one of the greatest scientific adventures of all time, reading out our own DNA instruction book. And I was saying no, because it sort of wasn't convenient. I was having a pretty good time where I was in Ann Arbor. It was risky. This might not work. I wasn't sure it really tapped into my skill set so much. I'd never run anything bigger than my lab. But gosh, you're going to walk away from that? This is one of those moments where a door opens that you don't expect, and you got to figure out, is this one for me? Or do I just walk through, or do I move on? And ultimately, I couldn't walk away. And so I said, okay. And I joined the NIH in April of 1993 to lead this effort, which had promised it was going to deliver a complete sequence of the human genome by 2005. Without really having any idea how that was going to be possible, because the technology to read out all those DNA bases was not in hand at all. On a good day, you might be able to read a few hundred letters of the DNA code. We had to read 3 billion. If you did the math, that probably meant once you really got up and going, maybe 1,000 bases a second. Okay, a big stretch there. A lot of work had to be done. But we kind of organized it. I had opportunity to tap into the best and brightest brains around, not just from the US, but from the rest of the world. This became an international project. And over the course of the next 10 years, bit by bit, with a whole variety of milestones that I'm not going to go into because it would take us all day, we did achieve goals that were previously thought unreachable bit by bit. And by 1996, 1997, started really seriously sequencing the human genome. By 2000, we had a draft, and by 2003, the finished version was announced. 2 years ahead of schedule, and happily for me, at a budget that ended up being about $400 million less than what the Congress had initially estimated. They don't see projects like that come along every day ahead of schedule and under budget. So it was a good thing to be able to say. But it was an amazing experience being part of this team effort. We got to announce this in an event, the draft that is, in the White House in the year 2000. But mostly I want you to look at this picture of the folks gathered there, in this case at the Cold Spring Harbor Laboratory, for one of our many genome meetings. Those are the leaders of the roughly 20 research institutions that were involved in this in a very substantive way. 2,500 scientists altogether, 6 countries, 13 years, all of them agreeing to put their shoulders to the same wheel and get the job done and not worry too much about who got the credit. And what an amazing output they managed to achieve in that timetable. And they also agreed, this is really critical, to give all the data away. That none of this information was going to be kept in any special hidden place. Every 24 hours, as soon as you had an assembly of DNA fragments that was more than a kilobase, it had to go up on the internet, and we lived up to that. And so people began to use it as soon as it was being produced, and no patents could be claimed on the parts that weren't already claimed by somebody. And we think that was a very important part of the success of this enterprise. So that's great, but I'm a physician. My dream was this is going to help people in the clinic. And the idea might happen that you would have the information, but not know what to do with it, as in this cartoon. So the next step was to try to figure out, how do we learn how to read DNA? And how do we understand differences between individuals? Because we don't all have exactly the same sequence. There's about 0.1% differences, and those matter. So a whole bunch of other projects got started rather immediately after the genome sequence was completed. Some of those continue to this day, basically adding value to the information. And all of these produce data sets that are public, and many of you are probably using their outputs right now for your own research. It's empowering for everybody, including people who are doing very basic science and people who are doing clinical applications. So that was fun, and I stuck with that, again, as an NIH Institute director, leading the National Human Genome Research Institute. But by 2008, I was like restless. I've done this for 15 years, sort of felt like maybe there's something else I need to do, but I don't know what it is. So I took a break. We had a nice party. The band got together. I've always enjoyed assembling various rock and roll bands at NIH, and we have a really good one right now called the Affordable Rock and Roll Act, although we've been a little bit out of gigs because of COVID. And I got to ride my motorcycle with my wife a bit more than usual. And the people who had wanted to say thank you gave me some support here so that I could design my own guitar with a double helix and mother of pearl on the fretboard. And of course, guitars have to have names. And so this is Rosalind, as in Rosalind Franklin. And I wrote a book about the way in which DNA science was really changing the perspective about medicine and introducing something that I called personalized medicine at that point, which is now more commonly called precision medicine. And then I tried to figure out, okay, now what do I do? Well, a few months went by, and then I got told what I was going to do in a wonderful way. And that is a call from the president, President Barack Obama, saying, I want you to come back to NIH, not to run the Genome Institute, but to run the whole NIH. How do you say no to that? And so back I came. This is building 1 on the NIH campus, where until COVID, I spent an awful lot of my time. Right now, I'm in my home office in Chevy Chase, about 4 miles from there, where I have been pretty much living for the last year and a half. And yeah, I'm still here because, to my surprise, after 8 years serving the Obama administration, I was asked to stay on for the Trump administration, and then more recently asked by Joe Biden to continue in this role. So I'm now serving my 3rd president. And may I say, it's wonderful that medical research is one of the few things that's still left that can be put into this space of not being entirely partisan. And I'm, I think, just about the only presidential appointee that has survived these transitions. And it's a great honor to continue to be able to do so. So a few things I could tell you about what we are doing at NIH, and then I want to take your questions. First of all, I think you know NIH pretty well, but just to remind you, we have an intramural program, which many of you are part of. That's our conducting research in our own laboratories. But most of our budget goes out there to universities, medical schools, hospitals, research institutions to support research, which we learn about because people send us grant applications. We put them through the most rigorous peer review system, and then we fund the ones that have the greatest promise. And of course, we support a lot of training. And so I hope we are supporting a lot of you in ways that are probably not quite as generous as you would like, but enough to help you be able to follow your path forward to making contributions at this remarkable moment in science. We also work hard on communicating information. And as one measure of our success, we always keep track of how many Nobel laureates were supported by NIH, and it goes up every year. Last fall, if you were paying attention, and I suspect you were, you might have noticed these two folks, Jennifer Doudna, who is one of the co-inventors of CRISPR-Cas, along with Emmanuel Charpentier. And then Harvey Alter from Physiology and Medicine, who's not only NIH supported, he's part of our intramural program. He's been at our clinical center now for almost 50 years and a wonderful contributory member of our biomedical community who made his observations about hepatitis C, which has now become not just a treatable, but a curable disease. So yes, we're proud of those NIH supported investigators, in this case, chemistry, physiology, and medicine. Other things that I've had the chance in these last almost 12 years as NIH director to nurture and encourage are a couple of big projects. Don't get me wrong, most of what NIH does is not big projects. It's investigator initiated, hypothesis driven research intramurally and extramurally, and that's the way it should be. But once in a while, something comes along where you could see, hey, if we got a team together, something that probably wouldn't just come across to us as a grant application, we could make things go faster. That's what the Genome Project was. That's what the Brain Initiative aims to do in a very different field, namely neuroscience, by trying to develop technologies that allow us to understand how circuits and networks do what they do to achieve very complicated human behaviors that all of us take for granted, but are based upon those 86 billion neurons between your ears and the way they interact with each other. And if we could understand all of this, the way in which this would influence our understanding and ultimately our treatment of conditions like depression or schizophrenia or Alzheimer's or PTSD would be enormously advantaged. So we're in the middle of this. It has a vision to 2025, and it will clearly go on beyond that. But already an amazing set of technologies and inferences have happened as a result of this special focus on bringing everybody together who has great ideas and then turning them loose. Another project I'm proud of because I think it is going to be pretty transformative in terms of our understanding, not only of how to treat disease, but how to prevent it, which ought to be a higher priority than it often has been. If we really want to understand all the individual factors that play out in terms of health or illness, and that includes genomics, but it includes environmental exposures and human behaviors, socioeconomic issues, all of those factors, then you want a very large study of individuals on which you have exquisitely detailed information. That's what All of Us is. This program, which aims, when it's fully enrolled, to have 1 million Americans participating, making information available, their electronic health records, answering a lot of surveys, giving blood samples, getting complete genome sequence as a result. All of them really as our partners. They're not human subjects. They're our partners. They're in this with us. And as you can see from the graph, we're already up to over 300,000 that have started enrollment. I'm one of them. If you want to be one of them, joinallofus.org is looking for you because we want to bring this forward in a way that has the broadest possible input of participants. And by the way, this also aims to have the greatest diversity of any program that NIH has ever mounted. And at the present time, 51% of the participants are racial and ethnic minorities, which is exactly what we had hoped to achieve. And that also includes diversity of socioeconomic status, educational level, rural versus urban, all of those things that sometimes get overlooked. This is going to be the kind of place where we can really understand health disparities in new ways. So all of us, come and join if you haven't already done so. And then, drinking from the fire hose, along comes COVID-19, and everything ends up being very much turned upside down as a result. It was early January 2020, as we at NIH became aware that what was happening in Wuhan, China might be a threat to the whole world, although I don't think any of us realized the potential that it might take 600,000 American lives and almost 4 million worldwide, which is the current count. But we knew we'd better get busy. And so almost immediately, our colleagues in the Vaccine Research Center began the process of designing a vaccine, which has just been an amazing story of how it has been possible for that effort now to lead us in the direction of what could be an end to this effort, although we still have a long way to go. A long way to go. Our COVID-19 efforts, which I'm not going to talk to you about unless people want to ask specific questions, have been in three different areas, the sort of three legs of the stool: diagnostics, therapeutics, and vaccines. For diagnostics, we have invested in the development of no less than 32 exciting new technologies that allow detection of the presence of the virus, including home testing, all of that coming about because of NIH and our RADx project. And we've been deeply engaged in assisting not only vaccines, but also therapeutics through a partnership with industry that I co-lead called Active. And there are a lot of lessons that one could point to in terms of what we might want to take out of this for the future of other areas of research, which is mostly what we're going to be doing. And if you're interested, this is a brief editorial that I wrote a couple months ago in Science Magazine. I've also tried to do what I can to try to cover topics about COVID in a way that would help educate the public about this. And I don't know if you're one of those who check out my blog every Tuesday and every Thursday, but if you do, you will have seen things like this coming across twice a week, most of them for the last year, about COVID, and trying to put forward new exciting things that I've seen in the scientific literature that I thought people might want to know about. Again, if you haven't subscribed, just go to the NIH directors page at nih.gov and you can see how to do that. And then there was another issue that I want to touch on, which came across in the year 2020. People will think of that as the year of COVID-19, but that wasn't the only thing that came across. We saw from COVID-19 just how difficult this experience is for communities that have been hit hard with health disparities for a long time. And then we saw the killing of George Floyd, and we had to recognize that, in fact, structural racism is a reality in the United States of America, as much as we would like to think otherwise, and that we, the biomedical research community, is not free of that history either. And as a result of that, we have worked very hard to put forward a new and bold approach about how we, the National Institutes of Health, might tackle these issues. And this is now written about in this commentary in Cell from earlier this month, which you've not seen, I would encourage you to look at. You will see this has a lot of authors. These are people who are co-chairs of a series of 5 different groups that have been working hard in a very unflinching way to say, what is NIH's contribution right now to structural racism and what could we do about it? And of course, that brings us into a space where we have to look in the mirror. And we have a lot of things that could be said about that. And again, there's a website here that may be able to give you more information if you want to read about this. A very important part of this is something called Unite, which is our way of bringing together our own staff to try to understand what could we do to make our workplace more welcoming to all participants, and what could we do with our research agenda to make it even more effective in addressing health inequities as well as health disparities. And we are moving that forward in the most aggressive way that NIH has ever managed. And we are determined that this is not a performative moment. This is real action oriented, not just words. And so track that with us, if you would. Finally, before I take your questions, one other thing that has just come along that you might or might not have seen. This is from last week. A proposal coming from President Biden, but one which all of us are warmly embracing. The idea of adding a new unit to NIH called ARPA-H, building on the success of DARPA, which is part of the Department of Defense, that has achieved a few things like the internet and GPS and self-driving cars and a bunch of other really remarkable things by having a process of tackling really bold projects in a very flexible, innovative way. The kind of thing that probably wouldn't happen in our standard grant portfolio, something that would not be appropriate for most of what we do, but in some instances could really move things along. So we'd be delighted to have this as a new part of NIH's future. It's being debated right now by the Congress to see whether they think it's justifiable to start in the next fiscal year, and we'll see how that comes out. So far, it sounds pretty positive. So if you haven't seen this, have a look. That may be a pathway into the future for some projects that otherwise would take a lot longer. Finally, a few lessons from the journey here, and these are my own sort of observations that you don't necessarily have to accept, just coming from my perspective. One of them was to do what I sort of didn't do when I narrowed very quickly into the field of chemistry and physics and decided to ignore everything else. It's good to keep your horizons wide open and see what's out there. You might discover there's something that's really exciting, and you wouldn't have known that if you'd already closed out that possibility. As part of that, nurture your curiosity, not just in the area you're currently working on, but across the board. Definitely for biomedical research, the era of being just descriptive and studying problems is pretty much passing away. We now are in the position to be able to do things with great quantitation. And that means collecting large data sets, many data sets that are available to you for free, and learning what to do with them. So if you have the chance to be given the skill sets of a coder, do that. I think every graduate student, every postdoc, if possible, ought to learn how to do things like code in Python and R in a way that would allow you to tackle problems that might otherwise just seem a little too complicated and out of reach. Find mentors who can advise you. We like to be asked to mentor trainees. I do. I don't know very many professors who don't feel the same way. This is like what we enjoy most of all, is a chance to try to share the excitement of science and to guide trainees over rough spots. And there are going to be rough spots. There always are. And we've been through them too. So don't be shy to ask people to become your mentors. And they'll stick with you. And they'll often give you wise advice at a time you need it most. Life is unpredictable. Your career path is unpredictable. If a new door opens for you, well, check it out. Maybe it's like not for you, but maybe it is. I'm, of course, thinking about this idea, leading the Human Genome Project, not a door I thought would ever open. If another one closes that you thought, oh, that's what I'm supposed to do, and somehow it didn't pan out, well, maybe that's just as well. Maybe something else is your new calling. If you get the chance, as you get to be more independent, and right now as a trainee, you have some limitations there in terms of exactly what project you're going to work on. But if you have some input into that, and later on you'll have a lot more, try to pick projects that are really important, really exciting, really fundamental, that if this works, I'm going to actually make a contribution that's going to change things. There's a lot of scientific projects that aren't quite that groundbreaking. They need to happen too. But if you have the chance, you only get to do this once, pick something that has that kind of fundamental, groundbreaking appeal to it. I think that's very much worth looking at. And likewise, I guess most of those groundbreaking projects are risky, so don't be afraid of taking risks. Don't expect any of you to travel a linear career pathway. I don't know hardly anyone in biomedical research who could say they've had one, at least not recently. Things are changing so quickly, and that's okay. Again, doors opening and closing. That means linearity is not likely to be your experience. And don't be surprised if your plan for the next 30 years turns out rather differently. And it is an amazing time to be a scientist. All of the things that are possible now that I would not have dreamed, you're going to take part in that. I'm a little jealous as you're embarking on this journey at a moment where some of the big questions are going to get answered, and you're going to be part of that. And wow, what a great time to be a scientist. Again, I understand COVID-19 didn't help much, but we're going to get through that. And in terms of the scientific potential, maybe it's even been enhanced by that, the scientific credibility based on the development of vaccines that are saving hundreds of thousands of lives. That hasn't hurt our scientific community either. And then have fun. This is not supposed to be just, put your head down and go forward. This is supposed to be an opportunity to work with others, build relationships with colleagues, and don't forget, most importantly, treasure your other relationships with your significant others, your families. Some of you who have children, those are not to be sacrificed on this particular basis. That is also a way in which you become a fully formed human being as well as a fully formed scientist. But have fun. This is an amazing privilege to be able to be part of some adventure that is as exciting and significant as what we all get to do. And then if somebody says to you, well, okay, what do you think you're going to be doing in the next 10 years? You probably get that question a lot. And you feel like, oh, I don't know how to answer that. Just quote this one back to them. This is the guy who wrote The Little Prince, one of my favorite books when I was growing up. And Antoine de Saint-Exupéry, in a different book, not The Little Prince, wrote this: "As for the future, your task is not to foresee it, but to enable it." That's probably true of biomedical research in general, but it's probably also true of your own pathway. Enable something exciting, even if you can't quite see where it's all going. With that, and with this reminder, and by the way, that's my Twitter handle if you want to join my 150,000 followers, and that's where you can go to be part of the twice weekly blog distribution. I'm going to stop sharing and see, oh my God, there's 41 questions. So I'm going to have to turn to Sharon to help me sort those out.
So I'm going to do my best to put some together and cover a variety of different topics. Many students pointed out that you have this unique role working as a scientist, yet in a political arena and an administrative capacity, and how you balance your rigor in science with the sort of messy truth of politics, and how you navigate that, especially now during COVID.
Whoa. Well, yes, it is messy for sure. I have tried my best, especially since I came to be NIH director, to avoid any kind of alignment with political perspectives or parties. I'm an independent. I've never signed up with any given party. I have political views, but I generally keep them to myself. And I think that's really important for medical research. As soon as we start as researchers to take political positions that are based on something other than science, then it's easy for somebody to start to disregard us. So I worked very hard. I have spent countless hours on Capitol Hill building relationships with members of Congress. And those relationships for NIH are really important in terms of sustaining what we are all doing. And is partly responsible for the fact that our budget has actually gone up by more than 40% in the last 6 years because of this strong support from both parties, both houses of the Congress. And I have lots of very well-informed leaders there that I have worked hard to respond to every question that they've ever had. I think that's our goal, is to avoid any risks that medical research becomes a partisan issue. It should not be. And it's actually not so hard to make that case. Everybody cares about health issue for themselves, their families, their constituents. And if NIH is the best solution we've got to those, then why wouldn't you want to support it? And I think that's basically the way it's played out.
Thank you. Along the same lines, Alexa Bailey from Brigham Young and Megan Elliott from USC, who's actually here this summer in NIA, both raised questions about science literacy and health literacy, and how COVID has probably been this awful example of difficulties in our country around science literacy, and whether you have thoughts about how individual scientists can help with that.
Yeah, it's a really important issue, Alexa. And I must say, from my perspective, it has been disheartening to see with something that is so important. It's not just an opinion, it's life and death, that it has been difficult to get the actual facts and evidence about what actions we should all take accepted by people. We do have an issue about science literacy in this country, and that's not just happened because of COVID, it's been there before. We also, I think, have a difficulty for many people in the public understanding statistical risk, a pretty simple concept, but readily misunderstood and often susceptible to overplaying risks that are actually extremely remote. And unfortunately, on top of that, all of those issues have now gotten mixed together with politics, and politics, which has often been fairly malignant in its contribution and has basically overshouted the scientific evidence that might have helped people make more rational decisions. When you look at where we are right now, where we have 3 vaccines approved by FDA that are safe and effective, and yet we have about 90 million people in the United States who have not yet signed up to take a single dose, even though those are free and now widely accessible. Talk about an indictment of our failure to communicate science in a way that actually got people to make rational decisions. There you have it. Scientists are in a special role here to be able to make that case. and we should not be shy to do so, but also being careful not to align ourselves with political perspectives. One of the people I've greatly enjoyed working with and admired in all of this is Dr. Kizmikia Corbett, who was one of the postdoc scientists working the Vaccine Research Center that is responsible for the fact that those mRNA vaccines came through. And Kizmikia happens also to be an African-American woman, and she has put herself out there repeatedly in multiple different settings to try to reach out to resistant communities and explain the facts about the vaccines. Now, not all of you can be Kizmikia Corbett, not all of you worked on the vaccines yourself, but you have access to information. Become ambassadors. Don't be shy to do so. Listen first before you start lecturing. I've learned that along the way, although sometimes I forget. But you have information, and you have information that can come across maybe as a way of screening out all the noise that's out there of information that's wrong. You're a scientist. You will have some respect for that if you figure out how best to interact with people who still have their minds open. So yeah, guess I'm charging all of you to be part of the solution.
We all need to open our ears more, hear more, and also try to talk more.
Yeah.
Calmly, though, and openly.
Calmly, yes. Yeah. Shouting doesn't work.
Doesn't work. And we have a lot of that going on in
we sure do
right now, too much. There's many questions about mentoring and mentorship. So Mauricio Flores, who's a grad student at Roswell Park Cancer Center, Paul Borgman, who's a med student at Florida State, Julia Versal, who's also a med student at Chicago, all point out that you mentioned the importance of mentorship. You talked about, times in your career where you had big decisions to make, and sort of who are some important mentors for you. And then the flip side, as a mentor to graduate students and physicians thinking about academic research, what advice would you give as a mentor?
Well, it is a really critical issue, and I've been fortunate to have those kinds of mentors, starting with my undergraduate mentor at the University of Virginia, who has been a lifelong friend, Carl Trindle, who I'm comfortable reaching out to at any time to catch up on things and to seek advice. And that is one of the important aspects, I think, is this is not something that you sort of do for a day or two and then you're over with it. Mentors can really be life partners for all of us, and we need that. And along the way, gosh, when I first came to NIH, I had no idea what I was doing. I came from academia. I didn't understand all this bureaucracy, and I was supposed to run an institute. My mentor, you may be interested to know, was Tony Fauci. Tony, as you know, our now best known infectious disease expert, had already been at NIH running an institute for 10 years when I got here, almost 30 years ago. And he gave me all kinds of really good advice about how to make this work in simple, straightforward ways. And we continue to do that for each other. Maybe it's gotten a little bit more symmetric as I got further along, but initially it was Tony helping me along. And that's, again, I was a little reluctant at first to go and ask for help because I didn't want to necessarily admit I needed it. And maybe you have that feeling sometimes too, like, no, I should be able to cope with this. Don't feel that way. Actually reach out and see what kinds of help you're able to get simply by saying you're interested in that. I'm particularly concerned, though, about people for whom mentoring may not come so naturally. Hey, I'm a white guy. I'm part of this network that happens pretty naturally in an academic environment. It wasn't hard for me to be able to do that kind of outreach. But if you're somebody from an underrepresented group, or even for women, although it's getting better, it's not where it should be, it may be a little bit more of a step forward. And maybe then I would want to say to all the potential mentors, hey, this is not just waiting for somebody to come and say, will you be my mentor, but offer yourself. If you're somebody who sees a promising young scientist, reach out and say, let me help you. And I think that is one of the greatest joys of being a scientist, is to have that opportunity. Sharon, you mentor hundreds of people, because I know many of them, and I don't know how you juggle all of that, but talk about a model for somebody who seems to always have just the right sense of what somebody needs to hear at a given moment. Sometimes it's a kick in the butt, sometimes it's a shoulder to cry on, sometimes it's both of those in the same day. You have instincts there that are obviously of great significance and importance.
Thank you. I would say that the thing is that I get as much from the people I mentor as I think they get from me. And you probably feel the same. You learn from your trainees and the people who come from you. And so it's really a satisfying back and forth relationship.
Absolutely.
Yeah. I should just make a plug. Next week, the OITE will offer a webinar, Maximizing Mentoring Relationships, which will talk about expanding mentoring networks and a little bit about dealing with and improving relationships with mentors you have. I'm going to change directions a little bit. Skyler Sung from Amherst, who is also at NHGRI this summer, and several other people, but I'm not finding the names right now, have pointed out that surely you've dealt with a lot of setback and disappointment. They've also pointed out that you must juggle a lot in your life, and asked whether you could talk a little bit about how you stay calm in the midst of setback and disappointment, and a little bit about how you find time for your family, friends, and taking care of yourself.
Well, Skyler, it's a good question. And medical research, as it is said, can be a selfish mistress that seems to expect more of you than you could possibly offer sometimes. And we need to really think about how to achieve that balance. And I'm probably not a great role model for that, although my intentions are generally pretty good. I'm fortunate that I have the most amazing life partner in my wife, Diane. She's a genetic counselor. So we have this area of science in common, which is nice that we can talk about it and understand each other. But she's highly motivated also to make the medical research arena successful. And she has been an amazing partner in everything we've done at NIH to create a sense that this is not just a faceless institution, but it's a place that cares about people and cares about families. That has been a great gift. I do find that, left to my own devices, I can get a little overstressed and overworked. And certainly COVID-19 has created that incredible intensity where I think for many weeks over the course of the last year and a half, I've been working 100 hours a week. And that's just not healthy. But when you have the sense that people's lives are at stake and every day counts, it's hard to say, okay, I got to take a break. This last week was the first time I took a little time off. And I went with my daughter, who's a physician in North Carolina, and my 3 grandkids, her kids, and my wife, and went to the beach in Oak Island, North Carolina. Yeah, I was working about half the time because it's pretty easy to do that remotely, but it was a little different. It was the first time I have seen those wonderful people in a year and a half. One of those grandkids, by the way, is currently a summer intern at NHGRI. So I was checking out whether he was really dedicated to the experience by how much time he was spending on the laptop during the week. But it was pretty good. Sellers, you did fine. That just is a reflection, I guess, of how much we all need this kind of support. There are other things that I do on a daily basis to try to keep myself grounded. One is I'm a person of faith. And I try every morning when I get up, usually about 5, to spend a little time in meditation, reflection, reading the Bible, praying. That just gets me sort of ready for the day. I'm also a musician of very amateur sort, but I enjoy that experience a lot. And if I'm at a point where I've got 10 minutes where I don't have to do something, I'm likely to jump out of this chair and go across to the grand piano and play some music just to kind of clear my brain out a little bit and get back into a different zone. And exercise, I'm a bit of a nut about that with weight training and cardio and biking on the weekends. Also really good for your mental health as well as your physical health. And there's that Harley Davidson motorcycle. Yeah, there's that too, which is also kind of a rush when you just need to get out of the house and have the wind blow around you.
It's interesting. I've heard you say a couple of times you don't feel like you're a very good role model in that regard, but the honest truth is you have some things you love to do and you make time. So maybe you're a good role model in really enjoying it in the moment, right?
I do enjoy it in the moment. You're
right. And knowing when you need it, because it's interesting. Every time you say that, then you come with these examples of things which seem so important. So maybe a good lesson there. There are a couple questions about systemic racism. And again, the questions are now so many of them, it's hard for me to find the names. But one started by making the observation that in many ways science started and still is a rather elitist endeavor in some ways. And so we have this history of exclusivity, and now we find ourselves really wanting to make dramatic change. And if you have thoughts about what individual scientists can do, what programs can do, what the NIH's responsibility is, sort of at the very personal level and then up to the very system-wide level.
Well, at the very personal level, we have to look in the mirror and recognize that our community, including all of us, including me, has not come across as welcoming to people of different backgrounds. That maybe we invite them in, but kind of not in a particularly warm way. And there doesn't seem to be the kind of natural mentoring and support that you would want to see in that circumstance. We've worked hard at NIH over the last few years with the leadership of Dr. Hannah Valentine, who was my first chief officer for scientific workforce diversity, to change that and actually to do intentional recruitment, not just of single investigators of backgrounds that are more different, but of a whole cohort, so that you had a natural cohort experience of arriving with another group of investigators who were similar to you in their life experiences, and then making sure the mentoring was there. That kind of organized program is necessary. But at the bottom line, Sharon, it's kind of up to all of us. Are we really interested in the conclusion that diversity is critical to enhance productivity? Or are we just thinking, well, that'd be a nice thing to have, but it doesn't matter that much? It really matters. And once you kind of recognize that, and that this is not only the moral choice, although it is, it's also the practical choice if you care about science, then how could you not make this one of your own personal priorities? But that's up to all of us. And then we have to have a recognition that it doesn't always go well. And there are people experiencing on a daily basis microaggressions or sometimes even frank racial discrimination. And we have to have a way to recognize that, have that pointed to, and have it stop. And we're putting in place some of those efforts now in our intramural program. And I think many universities are doing the same.
Yeah, I think we are living through a time of change in science where we've acknowledged some of the very best of what we do, and now looking at some of the things that we maybe have less to be proud of and want to change.
Yes.
So it's a positive time to see such a diverse group of students here. So I want to honor the international students and students who were immigrants. There are a lot of questions about this general feeling of being less welcomed in science, given some policies over the last years, and NIH funding decisions, and funding regulations about what they can and can't fund in terms of international students. And just your thought on the international and global nature of science and the importance of international trainees, I think would be important for us to hear.
Yeah. I think it was Louis Pasteur who said, science knows no one country. Science is supposed to be about humanity, and it is therefore by definition global and not domestic. Global health has been one of my highest priorities as NIH director, and it broke my heart to see the negatives that were getting piled on top of our international interactions in the past few years. And I hope we are now finding a way to start to turn those around, although there are still many difficulties in some areas. Yeah, I do want to reach out to the trainees from other countries who have not felt always as welcome as I would want them to be. You're such an important part of our talent pool. The reason that US biomedical research has been successful, that NIH has been able to support such great science, a lot of that is because of immigrants, foreign nationals who come to our shores and carry out amazing work, and many of them decide to stay, and we are absolutely honored and delighted that you do. We are constrained at NIH by what we can legally do, and the Congress makes those decisions about what kinds of awards we can give to people who are not citizens. When we get the chance, we try to bend those rules a bit. The K-99 is a good example, this sort of award that allows a postdoc to move into a tenure track position where we were able to get that approved for non-citizens. And that was a bit of a heavy lift, but we will continue to try to do what we can. And I think we now have a much friendlier administration towards such issues. So we'll work in every way we can to build on that while we have the chance. But yeah, I'm glad the questions come up. Certainly I've had many foreign nationals in my own research lab, and I know that is a special additional stress that falls down on your shoulders.
So I'm going to honor, there's a lot of questions from high school students, which excites me a lot to see. And so I'm going to honor sort of the future of science by asking the final question from one of the high school students who's a part of the High Step program here at NIH. And he asked a little bit about what NIH was doing in the realm of cancer biology, but there were several other questions from high school students about what you think the most exciting areas of science are going to be for students, right, who are just now starting out. They can factor in a little bit of a vision of the future. And I thought I'd put those together because of the President's Cancer Moonshot, which does give this feeling of futuristic science. And you want to just give us your thoughts about the excitement coming ahead?
Sure, I'll try to riff on that a little bit. Well, there is so much excitement. And if I picked one thing, it would be really doing an injustice to all the rest. So I'll just rattle off a few. Certainly cancer, yes. I mean, look what we have learned about the genomic basis of cancer. And now look what we're learning about cancer and the immune system with immunotherapy coming forward in ways that are breathtaking in some circumstances and looking for a way to get even better, working for solid tumors. So yeah, that's a place of great excitement, great potential. In the basic science arena, the idea that we could actually study single cells this is a revolution. We've not been able until about 5 years ago to really understand what's happening in a tissue, a human tissue, other than a chunk of tissue, which we knew had all sorts of heterogeneous cells. And now we can query each cell. What are you doing? And even what are you doing relative to the neighbor that's next to you, the sort of spatial transcriptomics. All of that is such cool stuff. We have technologies now that are both incredibly powerful for basic science, but also for clinical application. And here I'm thinking of CRISPR-Cas and the gene editing revolution. Again, back to why I was a couple minutes late coming to this. That's what we're proposing to do, to cure a rare genetic disease by in vivo administration of a gene editor. Wow, if that works, that's just so transformative. And there are thousands of other diseases for which the same approach could be taken. So how could you not get excited about that? We're curing sickle cell with that approach. And we need to figure out how to do that even more broadly. And then it's neuroscience. I got to mention neuroscience because it is the time of human history where I think we have the potential of really understanding that most complicated structure in the known universe, which you have between your ears, the human brain. Just amazing in its complexity, but maybe we'll even be able to figure out some of the real fundamental issues about how it works. Like, how do you lay down a memory and retrieve it? We're going to figure that out. How do you do all kinds of complicated things in terms of motor function, in terms of sensory input? We're going to figure that out. That would be fun to be part of, to be sure, especially if it leads us to lots of insights about neurological disorders, which of course can take the joy out of life for an awful lot of people and shorten way too many lives. So yeah, I could go on and on about technology, artificial intelligence, machine learning. Yeah, make me stop. But there is no shortage of fantastic areas of science to plunge into. Just pick your path carefully, find a good mentor, work hard, but have fun. You'll be fine.
Great, thank you. What a great place to end. For those of you taking part in the Resilient Scientist series, we start a little late today to give time for this, but we'll be back on Zoom at 2:30. Please join me in thanking Dr. Collins. I know he won't hear normally in the room. He would have a chance to hear your appreciation, but Francis, thank you so very, very much for all you do on behalf of all of us. Thank you.
Thanks, Sharon, for everything you do. And it was a pleasure to have a chance to talk to all the trainees.
Thank you. Be safe, everyone. We'll see you soon.